Okay. Okay, so thank you all for coming. So as Dr. Freebos mentioned, I will be providing a prelude to my dissertation defense, which we are currently planning for next spring. And one of the major foci in the Freebos lab over the past few years has been predictive modeling of lung cancer therapy and survival based on metabolomic data. And as a part of this effort, we have been developing a methodology to calibrate a mechanistic model of tumor growth to patient-specific metabolomic data for therapy prediction. So cancer is a complex disease, and the physical processes that drive tumor growth and make individual tumors different from one another are not yet fully understood. These processes are interrelated and occur across a wide range of spatial and temporal scales. For several years, a monumental effort has taken place to develop a multi-scale model of solid tumor growth to better replicate what is observed in vivo and build a foundation for in silico experiments. In the model, tumor tissue receives nutrients and growth is driven by pressure with an associated extracellular matrix. Hypoxia and necrosis can occur due to the limited diffusion capacity of oxygen and other nutrients, resulting in a heterogeneous tumor spheroid, where proliferating tissue is represented in red, hypoxia in blue, and necrosis in brown. To more accurately represent in vivo conditions, tumor vasculature is modeled by a process of angiogenesis, where hypoxic tumor tissue releases tumor angiogenic factor, and a neovasculature is formed and migrates towards regions of hypoxia, delivering nutrients such as oxygen. An increase in nutrient concentration drives the pressure gradient, which stimulates tumor growth. Within the simulation domain, there is a two-dimensional grid where the numerical computation takes place. This illustration shows how the migration of endothelial cells from a parent vessel in the host tissue can form capillary sprouts. At each node, a sprout tip can migrate in 3 possible directions. Each of these directions is associated with a probability based on the sum of functions of chemotaxis and haptotaxis, which are dependent on the concentrations of tumor angiogenic factor and fibronectin. Once a capillary has been established in the domain, blood flow is modeled as a non-Newtonian fluid, dependent on viscosity, hematocrit, vessel radius, and pressure. The vessels adapt their radius to maintain a constant level of wall shear stress, described as a logarithmic law, which is a function of viscosity, hematocrit, vessel radius, and flow. In more recent work, a host vasculature surrounding the initial spheroid is present, from which a neovasculature arises. These representative images show the development of the neovasculature and tumor growth over 10.5 days. There have been numerous publications where this type of model has been applied, and today we'll focus on just a few, including an evaluation of drug-loaded nanoparticle cytotoxicity in simulated vascularized lesions, and how differences in tumor tissue heterogeneity affect this type of therapy. I'll also show an immune system model of tumor-associated macrophages, which was developed using a version of this model. And we also investigated how macrophages and different extracellular matrix characteristics influence tumor growth following liver injury. We've also been developing a methodology to calibrate the tumor growth model based on patient tumor-derived metabolomic data, which we hypothesize will serve as the basis for a predictive modeling workflow for lung cancer therapy outcomes. As part of an effort to better understand how tumor vasculature may affect cancer nanotherapy, this study focused on an interdisciplinary approach by utilizing data from cytotoxicity assays and simulations of vascularized lesions. To help replicate what is observed in vitro, avascular spheroids can be simulated by switching off the process of angiogenesis. 2- and 3-layer gold core nanoparticles were synthesized and loaded with either paclitaxel or cisplatin, so their individual efficacy could be measured. The gold nanoparticles are made with a citrate stabilizer and a layer of hexadecane thiol to provide a hydrophobic core. A layer of phosphatidylcholine promotes water solubility, and 3-layer particles contain an additional layer made of the components of high-density lipoprotein, which improves targeting of the tumor tissue. The efficacy of cisplatin and paclitaxel as free drug and formulated as nanoparticles were evaluated in 3 different cell lines, including A549, H358, and PC9. Drug efficacy was measured as cell viability as a fraction of control on the y-axis and drug concentration AUC on the x-axis. For each cell line and drug type, the 2-layer and 3-layer nanoparticles, represented by the triangles and squares, were more effective at reducing cell viability in 3-dimensional spheroids compared to free drug, which is represented by the solid black curves. Another important takeaway is that cell viability was much lower in monolayer, meaning that the drug was overpowered in traditional cell culture, while the spheroids are likely providing a more realistic replication of in vivo conditions. In the model, oxygen and nutrient transport is accomplished through a discrete vasculature by a steady-state diffusion equation with pressure-dependent extravasation from the vessels. Nanoparticle transport is modeled by steady-state diffusion, where the extravasation from the vasculature is concentration-dependent. Drug is then released from nanoparticles and diffused into the surrounding tissue. Tumor growth follows Darcy's law and is associated with the rate of volume change. Drug effect is modeled by including a loss term in the pressure equation, and cisplatin and paclitaxel are both cell cycle dependent drugs, so only the proliferating region of the tumor is affected. These representative images of simulation results show the spatial distribution of nanoparticles and drug within the domain of a vascularized lesion over a 24-hour period after the initialization of treatment. Paclitaxel on the left has a relatively slow release rate and continues to increase in concentration over 12.5 hours, while cisplatin has a much faster release rate and a much faster decay. As a result, the paclitaxel-loaded nanoparticles were a more efficacious therapy. Drug release rates were fitted to previously obtained experimental data and calibrated in the model. The cumulative release of paclitaxel increases linearly for several days, and for comparison, cisplatin increases rapidly and plateaus within just a few hours. Although the nanoparticle decay rate was the same between both formulations, paclitaxel clearly results in greater tumor regression over time, as depicted by the fine dashed curve. To continue with this effort, we investigated how tumor tissue heterogeneity, driven by differences in the necrotic and hypoxic oxygen thresholds, affected nanotherapy response in vascularized lesions. Differences in tissue heterogeneity were simulated, resulting in differences in vasculature, represented here by the generalized cases of very low, low, medium, and high heterogeneity. In these 4 cases, we observed differences in nanoparticle and cisplatin concentration over time. These representative images show the proliferating, just to reiterate, the proliferating region in red, the hypoxic tissue in blue, and necrotic tissue in brown. Although the decay rates in the magnitude of the drug effect were held constant across these 4 cases, the distribution of nanoparticles in drug changes with differences in neovasculature and tumor necrosis. Drug treatment was simulated as a bolus injection and was varied from a 24-hour IC50 on the high end to a 96-hour IC50 on the low end of drug strength. The lowest heterogeneity tumor represented by the light gray bar, and the highest heterogeneity tumor is represented by the black bar. Interestingly, the area under the curve for intratumoral nanoparticles was highest in the low heterogeneity case, regardless of the drug strength. While the drug effect was measured as the minimum tumor radius achieved after bolus injection. We can see that tumor heterogeneity has the strongest effect on tumor radius, with the strongest drug concentration of a 24-hour IC50, where drug was most effective in the very low case and least effective in the medium case. However, at weaker concentrations, drug tended to be least effective in the very low case, although the differences here are very small. From this study, we concluded that drug efficacy has a nonlinear relationship with concentration and tissue heterogeneity. Tumor response becomes more insensitive to variation in drug strength as this strength increases or as tissue heterogeneity increases. This study represents a first step towards the development of a principled approach for predicting nanotherapy efficacy using patient tumor-specific characteristics. Limitations include a simplified pharmacokinetic and pharmacodynamic model. That leads us directly into another publication, which involves a more advanced pharmacokinetic pharmacodynamic model to simulate nanotherapy. Here, nanoparticle and drug transport are modeled with multi-compartmental kinetics. Nanoparticles diffuse through extracellular and cytosolic compartments, while cisplatin diffusion includes an additional DNA-bound compartment. Each directional compartment transfer is associated with a unique transfer rate, K. Nanoparticle aggregation was also simulated by modeling aggregation blocks of different sizes, where nanoparticles in the surrounding area are stacked and concentrated onto a single node. Drug effect is modeled by the cumulative DNA-bound cisplatin multiplied by a scaling factor to achieve the desired effect. These representative images show nanoparticle and drug spatial distribution across the tumor boundary within host tissue and within all of the compartments. A nanoparticle bolus injection was simulated using a range of cytosolic transfer rates from 0 to 50 in non-dimensionalized units. A low heterogeneity tumors have a decreased abundance of both nanoparticles and cisplatin across all compartments. These images show the distribution of nanoparticles in drug with increasing nanoparticle aggregation after the drug has taken effect. Nanoparticle aggregation results in localized pockets of drug, which also seem to affect the regions of eliminated tumor tissue. Affected regions of tumor tissue become less uniform with increasing nanoparticle aggregation, and this effect is more pronounced in the high heterogeneity tumors compared to the very low heterogeneity tumors. Here we can see that increasing nanoparticle cytosolic transfer rate in panels A, B, and C with the lowest represented by the black bars and the highest represented by the light gray bars. Increased drug concentration found in the cytosolic and DNA-bound compartments, although it had no effect on the extracellular drug concentration. Increasing nanoparticle aggregation, panels D, E, and F, with the lowest aggregation as the stripe bars and greatest as the black bars, greatly increased extracellular drug concentration across all tumor heterogeneity cases. Interestingly, a different pattern was observed in the cytosolic and DNA-bound compartments, where minimal nanoparticle aggregation resulted in the highest drug concentration for the high heterogeneity case, while medium aggregation produced the highest drug concentration for the medium heterogeneity case. The minimum tumor radius as the fraction of initial was affected by both cytosolic transfer rate and nanoparticle aggregation. The strongest drug effect was achieved with the highest cytosolic transfer rate of 50 in the high heterogeneity case, while the lowest drug effect was observed in the very low case with a cytosolic transfer coefficient of 5. Minimal nanoparticle aggregation generally increased drug effectiveness across all levels of heterogeneity, while maximum aggregation generally decreased drug effectiveness across all levels of heterogeneity. In this study, we concluded that although increasing tissue heterogeneity had no effect on extracellular drug concentration, cytosolic and DNA-bound drug concentration was increased. Nanoparticle extracellular drug release was critical for achieving a higher concentration of cytosolic and DNA-bound drug, especially for low heterogeneity tissue. Also, nanoparticle aggregation may be beneficial for increasing nanotherapy efficacy, depending on the drug used and the characteristics of the tumor tissue and the associated vasculature. However, the pharmacodynamic model that we used here does not account for drug resistance mechanisms, and the multi-compartmental kinetic parameters are generalized and not calibrated to specific nanoparticle and drug combinations. Additionally, the tumor growth parameters were not calibrated to patient-specific cases. This is something we addressed in more recent work, as I will show you shortly. One of the more recent additions to this modeling platform was the development of an immune system model simulating tumor-associated macrophages. So cytokine expression is simulated by a diffusion of factors from tumor tissue, which influence macrophage polarization. Tie2 macrophages release the cytokine interleukin 10 or IL-10, which promotes the polarization of monocytes into type 2 macrophages. So in the model, macrophages are discrete units in the domain of the system, and they migrate semi-stochastically towards a chemoattractant gradient, which is also produced by hypoxic tumor tissue. Once a monocyte is spawned in the domain, it has the option to differentiate based on the concentration of various cytokines, as calibrated to experimental data. Type 1 macrophages, or M1s, are tumoricidal through the cytotoxic effect of nitric oxide, while type 2 macrophages, or M2s, are tumorigenic, and they accomplish this by enhancing the proliferation rate and lowering the viable oxygen threshold. Type 2 macrophages utilize angiopoietin-2 as the chemoattractant, which is released from the vasculature, and they also promote neovessel sprouting, thereby enhancing tumor growth. Various tumor growth scenarios were compared over time, including the presence of these macrophage types in all combinations. The extent of vascularization relative to the tumor growth over time is represented in this figure. Combining M1s and M2s with with Tie2 macrophages generally increased the vascular volume to tumor volume ratio, and inclusion of Tie2 macrophages on their own resulted in the largest increase, indicated by this dotted line with empty circles. The M1 subtype plus Tie2 macrophage subtypes combined resulted in the largest total tumor vascular area after 13 days of growth. However, the M2 macrophages on their own resulted in the largest tumor radius despite having less vasculature. Taken together, these results indicate that there is a complex relationship between these macrophage subtypes and very different tumor growth patterns observed with various macrophage populations. We also applied a similar modeling platform for evaluating how macrophages and different extracellular matrix characteristics influence tumor growth following liver injury. Here we simulated primary tumors and compared them to metastatic tumors in which the host vasculature is denser and the necrotic oxygen threshold is lower in the metastatic case. We also varied the extracellular matrix physical properties by increasing the production rate and decreasing the degradation rate in a transitional ECM as compared to a control ECM. The cytokines TNF alpha and TGF beta were diffused from tumor tissue and influenced macrophage polarization to the M1 and M2 subtypes, respectively. Levels of these simulated cytokines were determined from experimental data. We evaluated 8 different cases in silico to observe the rate of tumor growth over a period of 13 simulated days. With regards to macrophages, there were 2 control cases with undifferentiated macrophages or monocytes only, as the light gray bar and black bars. The inclusion of type 1 and type 2 macrophages had no significant effect in the primary tumor compared to the controls, but it significantly lowered tumor growth in the metastatic tumor across both the control ECM and transitional ECM. The evolution of macrophage subtype populations over time shows that the primary tumor on both ECM constructs exhibited a more balanced proportion of M1 to M2 subtypes, while the M1 subtype dominated the macrophages in the metastatic tumor. From this, we can conclude that there's a complex relationship between extracellular matrix characteristics, vascular density, tumor hypoxia, and macrophage polarization due to cytokine signaling, and how all of these things come together to affect tumor growth over time. So this type of modeling platform shows a lot of promise for future work, including more specific calibration of ECM physical characteristics and immune cell information obtained from human patients. So the tumor growth model has proven useful for simulating a variety of cancer types. And for my dissertation work, we wanted to focus on an application in the study of lung cancer. So lung cancer accounts for about 13% of all new cancer diagnoses. And although the mortality rate for lung cancer has been steadily declining over the past couple of decades, as indicated by the brown circles in this chart, the 5-year survival rate for advanced stage lung cancer is only about 20%, making it the most lethal form of cancer. There are a variety of treatment options for patients with lung cancer. Only about 17% of patients have a surgical resection, and this is because most patients diagnosed with lung cancer are already at an advanced stage in which surgery is not a viable option. Radiotherapy is a targeted form of therapy that's often used in combination with surgery. And the third option is systemic therapy, which can be subcategorized as either targeted or untargeted. Chemotherapy is considered untargeted in that it affects all cells in the body and remains the standard of care for advanced lung cancer patients. A major problem with chemotherapy is the dose-limiting toxicity. The effectiveness of chemotherapy is limited partly due to the challenge of determining patient-specific tumor characteristics and predicting how each patient will respond to established drug regimens. Outside of histological subtypes and a few genetic markers, there is no proven methodology to determine which combination of drugs will be most effective during first-line therapy. So this problem presents a need for a more predictive methodology to determine patient-specific tissue behavior in the presence of first-line therapy. So I will briefly go through one of our publications focused on using machine learning for predictive modeling of lung cancer therapy. In collaboration with Norton Hospital and Dr. Van Berkel at the University of Louisville Hospital, we obtained tumor core biopsies from 39 patients, of which 34 had pathological staging and 23 had therapy response information. Patient response was evaluated clinically and categorized as either complete response, partial response, stable disease, or progressive disease. And we classified the responses as complete response and partial response versus stable disease and progressive disease. So once the biopsies are obtained, the tumor tissue is incubated in carbon-13 glucose media for 24 hours, homogenized, and then a liquid-liquid metabolite extraction is performed. The polar layer is isolated and lyophilized before a mass spec analysis. The data pre-processing is performed to combine positive and negative ion modes and also to transform and center the data. And we also impute missing values and then categorize the samples based on either response or staging. Then we proceeded with statistical analysis and machine learning. To help predict patient classifications, we used two different methods of feature selection as part of the machine learning workflow, either by random forest or partial least squares discriminant analysis, or PLSDA. Then the predictive models were trained and validated with k-fold cross-validation. In a partial least squares discriminant analysis score plot, right here, we can see a clear separation between disease control and progressive disease groups, indicating that the two groups of samples can be organized by the patterns found in their metabolomics data sets. The top performing machine learning algorithm was a support vector machine, or SVM, using partial least squares for feature selection with an AUROC of 0.97. The median overall survival for progressive disease patients was 17 months shorter than disease control, while progression-free survival was 34 months shorter. So several machine learning algorithms have methods for determining variable importance based on the feature's contribution for informing the model's predictions. To help filter out the noise from the data set, we employed forward feature selection based on the variable importance scores from partial least squares. As indicated by the dark orange bars, the support vector machine used the top 12 metabolites to create the optimal predictive model. Overall, in this study, 66 metabolic biomarkers were identified from patient tissue samples between negative and positive ion modes from patient tissue after metabolite extraction. A log transformation normalized the data set well to prepare it for analysis, and feature selection proved to be a powerful method for filtering the noise out of the data set. A support vector machine resulted in outstanding prediction accuracy for disease control versus progressive disease with repeated cross-validation. So the biggest limitation of this study was the small sample size, which did not allow for stratification of the patient population based on therapy type and did not allow for an external test set validation. Using the metabolomics data set from this paper and the calculated variable importance, we wanted to investigate if our mechanistic tumor model could predict patient outcomes. So one of our current studies, which is in revision, aims to develop a novel mechanistic modeling platform for simulating patient-specific tumors calibrated to metabolomics data. We first established a baseline case which represents the average necrosis, hypoxia, and growth rate of the average lung cancer tumor. And then we established ranges for 7 different parameters, including the tumor proliferation constant, lactic acid production rate constant, which affects tumor aggressiveness, and sorry about that. Can you see my screen still?
Yes.
Yep.
Yep.
Oh, sorry. Yeah, it looked like Teams disconnected there for a second. Okay, I'll keep going. So oh, so yeah, something weird's going on there. I'm getting okay. So we established ranges for 7 different parameters, including the tumor proliferation constant, the lactic acid production rate constant, which affects tumor aggressiveness, the monocyte production rate, and the polarization constants for type 1 and type 2 macrophages. We also calibrated the drug effect and the rate of by angiogenesis. Before interpolating the values of each of these parameters, we first established a baseline case, which represents the typical lung cancer tumor using experimental data obtained from needle biopsies. And we also used hypoxia PET imaging and a 3-dimensional lung xenograft model. To calibrate the model, we fitted curves to match measured tumor metrics at a specific time point by adjusting the oxygen thresholds for hypoxia and necrosis and the tumor proliferation rate. In panels A and B, we fitted the proportion of necrotic and hypoxic tissue at 400 hours. And in panel C, the tumor proliferation rate was calibrated. To create a system which is flexible enough to simulate a variety of patient-specific tumor characteristics, so we determined ranges for several different parameters by measuring tumor growth metrics at a specified time point and fitting the data to curves. So after each model parameter was varied to establish calibration curves, the curves were mapped to a new y-axis on a scale from 0 to 1. So given a value of p sub y, the parameter values can be interpolated by then mapping to the x-axis. Using either machine learning or a statistical analysis, we assign a weight coefficient to each metabolite to determine its concentration to the parameter calculation. So far, we have tried using partial least square the dependent variable to provide weight to each metabolite. Then we rescale each transform metabolite intensity value to a positive scale. The metabolite weight coefficients are rescaled relevant to the sum of all coefficients for that given parameter. And the desired patient
Hunter, you're going in and out. I don't know whether
the minimum values for the parameter are calculated, and then
Hunter?
the hello.
Yeah, you're kind of
Hi.
little. Yeah, I think you might want to cut your video feed. Maybe that might help a little. I'm just a suggestion.
Okay, is that better?
Yep.
Okay, sure. So basically, the value of the parameter is equal to the x coordinate responding to p sub y. So at this point, we have a variety of parameters to calibrate, and they need to be linked directly to the data. So we searched the literature to find connections between specific metabolites and model parameters based on cancer biology. And in this table, the first column of values represents the transformed and rescaled metabolite intensity values for a particular sample. And the values in the parameter columns represent the coefficients which we multiply with the intensity values. Once these values are multiplied, they are summed down each row, and the sum of these multiplied values represents the location on the y-axis of the respective calibration curve, from which we can map to the x-axis to find the actual parameter value. Values of 0 here in this table indicate that the metabolite should have no effect on that parameter according to current biological knowledge. So metabolites highlighted in green here are believed to promote tumor control, and metabolites in pink are thought to promote tumor progression. The direction in which each metabolite affects the model parameters, either by increasing or decreasing the model parameter, is determined by this. So we simulated 4 cases representing high baseline and low parameter values, and included a representative patient-specific sample case whose parameter values were specified in the previous table. So after the specified growth period of 400 hours, the sample case followed a consistent pattern regarding tumor radius, tumor vessel surface area, and proliferating tissue fraction. But it had a hypoxic tissue fraction lower than the high and baseline cases, and had the highest necrotic fraction out of all the cases, highlighting the nonlinear nature of the system. We also simulated a bolus injection of cisplatin between responders and non-responders. The data for these patients was borrowed from our previous publication, where therapy response was categorized according to the RESIST guideline, where patients in complete response and partial response categories were grouped as responders, and patients in stable disease and progressive disease categories were grouped as non-responders. As you can see, the model was able to differentiate responders and non-responders based on a significant difference in the fraction of initial tumor size at the beginning stages of therapy. So this study sets up a framework for calibrating patient-specific tumor simulations by linking metabolomic data to tissue scale tumor behavior, with the ultimate goal of simulating therapeutic agent efficacy prior to patient administration. We have shown that the modeling platform presented is flexible enough to showcase differences in chemotherapy response after calibrating parameters to patient-specific metabolomic data, and is consistent with clinically measured results. One limitation of this approach is that the metabolic signatures detected in other studies may differ depending on experimental techniques. So the association of particular metabolites to model parameters also depends on state-of-the-art biological knowledge and mass spectrometry techniques. However, this could be refined over time. Additionally, the calibration of the metabolites associated with each parameter should be evaluated more thoroughly. So we have 2 final manuscripts planned for the remainder of my dissertation work. The first involves the prediction of lung cancer therapy response and validating the results with clinical measurements using the modeling platform presented in the previous paper. Objective number 1 is to map metabolic feature spaces to model outcome spaces and find the optimal combination of metabolites affecting each parameter. And this will involve a combination of data mining and machine learning based modeling of simulation outputs from parameter ranges. Objective number 2 is to validate the model predictions by comparing them to the true clinical outcomes. Objective number 3 involves simulating a variety of chemotherapy regimens to determine the optimal drug nanoparticle combinations for each patient. And objective number 4 is to hypothesize explanatory mechanisms behind the response classifications to specific therapy regimens based on the simulation outputs. And there are a variety of variables we could potentially explore for that purpose. So traditionally, machine learning and mechanistic modeling are considered separate fields of study, but when combined, there is the potential to develop a more powerful predictive modeling framework. So mechanistic modeling can provide mechanistic insights by exploring feature spaces, and it can also be used to supplement sparse training data, while machine learning can help to identify parameter values based on training data and can act as a surrogate model to decrease computational cost and increase throughput during parameter sensitivity analysis. So for the final part of my dissertation, we are interested in investigating the potential for a machine learning algorithm to act as a surrogate model of the mechanistic tumor growth model. And a promising candidate is a generative adversarial network, or a GAN, which is a special type of neural network architecture composed of convolutional neural networks. It is often described as a generator and a discriminator competing in a two-player minimax game. So simultaneously, the parameters for the generator and the discriminator are adjusted until the discriminator is no longer able to distinguish real data from fake data created by the generator. GANs have been used for image generation, such as creating handwritten digits, faces, and recognizable images of many ordinary objects. In theory, one should be able to train a GAN as a surrogate model based on spatiotemporal simulation outputs. And the basic idea of a surrogate model is to provide sufficient training data for a machine learning algorithm, which is then capable of generating outputs similar to the mechanistic model outputs. So we hypothesized that a GPU accelerated GAN as a surrogate model for the tumor growth models can reduce computational cost and allow for high throughput patient therapy response predictions. The issue now is that the tumor growth model is CPU bound, and it would likely be too difficult and infeasible to implement GPU acceleration in the program directly by using something like CUDA in the C++ source code. So by utilizing a surrogate model, we would be able to take advantage of the TensorFlow package and the Keras API. In theory, the simulation runtime could be decreased from several days to only hours or minutes, and GPU clusters and advances in GPU technology over time offer the potential for scalability. So objective number 1 is to produce a set of training data from the mechanistic model simulations. And these will include simulations covering a wide range of growth and drug effect parameters. Objective number 2 involves developing a state-of-the-art neural network as a GAN with TensorFlow and Keras in Python, and generalize it to be trainable on outputs from multiple spatiotemporal mechanistic tumor models. And in objective number 3, we will perform high throughput sensitivity analyses of mechanistic model parameters by Monte Carlo methods with the goal of generating feature spaces where specific outcome classifications are likely to reside. So one of the most exciting aspects of this type of predictive modeling framework is the flexibility and the potential for expansion. So this type of modeling platform is primarily concerned with events at the tissue scale, but in theory, it could be coupled with other types of models. So I envision a long-term project which may involve the utilization of a model of metabolic networks, which would allow for fine-tuning the model parameters from metabolomics more directly. There's also the potential to expand the experimental approach by including experiments like immunohistochemistry, transcriptomics, or flow cytometry. And it would be interesting to see what other types of data we could gather from these patients and use them to calibrate certain model parameters. With a large enough patient population, we could stratify the patients based on specific therapy regimens, which would be useful when validating the model predictions. And longer term, we could also test how different concentrations of metabolites are related to tumor behavior with a more targeted experimental approach, which would increase our confidence in how the metabolite levels are linked to the specific model parameters. And we have already developed a lot of software for analyzing data and making predictions for cancer patients, and we could potentially integrate the tumor growth model into a convenient software package for others to use. And I imagine this as some sort of a web-based tool geared towards use by clinicians. And most likely we are still a ways off from something like this, seeing clinical application, but it is an exciting idea nonetheless. So we thank everyone who was involved in the generation of data and who helped with data analysis for these manuscripts, including several members of the Freebos lab. Dr. Lowengrub was one of the masterminds behind the tumor model, and we thank him for his contribution for helping to lay the mathematical foundation of the model. Dr. Van Berkel has been instrumental in helping us obtain lung cancer tissue samples from which we can calibrate the model to patient-specific cases. And of course, thank you to my committee members for their dedication and advice. And we thank the Department of Bioengineering, Department of Pharmacology and Toxicology, and funding from the NIH. And thank you everyone for your patience, and I'll take any questions.
All right, thank you, Hunter. We have some time for questions. Are there any questions? Please either raise your hand or you can type in your questions in the chat box. Hunter, I have a question. Is there any particular metabolite that you're kind of looking into further?
So we are not looking into any particular metabolites in an experimental sense, although there is the potential to do that. But from the modeling perspective, we're less interested in what one particular metabolite does, and we're more interested in how these metabolomics data sets as a whole can be used to calibrate the model parameters. There are a few metabolites that have been popping up as important for making predictions, and those include like pyruvate, isoleucine, and glutamine, and a couple of others. And there's some literature out there that describes how they're involved in cancer biology, but for a lot of these metabolites, we just don't know because we're doing an untargeted analysis. We would need a very targeted experimental approach to elucidate mechanism. And that's not exactly what we're doing here, but someone could potentially do that. So.
Thank you. Are there any questions from the audience? I must say a lot of it just kind of went over my head. It's a lot of mathematical modeling, and I must say I had a difficulty following the content, but I can see that you've been very productive in publishing papers, and also a couple of manuscripts in preparation. So.
Well, I apologize for that if it was confusing, but in the spring, we plan on my dissertation defense, and hopefully during that seminar, I'll be able to clear up a few things. It's still kind of diffuse right now, but in that seminar, I'll pull everything together and try to make a big picture of all the work that we've done. So.
Sounds good. I look forward to it. All right, thank you so much, Hunter, and thank you, Dr. Fribos. So next week we're going back, the hybrid, right? So there will be in person, and also you'll be able to join the seminar virtually as well. So hope to see you back here next week. All right, thank you.
All right, thank you.
Thank you.
Yes.